or what we call them the molecules of life. And these are all, we call them organic molecules. Why organic? Because any organic molecule, it must contain carbon together with hydrogen. So if you find carbon and hydrogen together in a molecule, so this is organic immediately. Inorganic, it does not contain both of them together. Let me go to the classification of these organic molecules, or they are classified into carbohydrates, fats and oils, or let me call them fats only now, and of course proteins. And there are other organic molecules that we have to know, of course. They are nucleic acids, and these are the DNA and RNA. These are acids that are found in the nucleus. All of them, the main components are carbon together with hydrogen. They must be found because, as I just said, they are organic molecules, so they must contain both of them. Now, for carbohydrates, we add oxygen. For fats and oils, we'll add oxygen as well. In proteins, we add oxygen and nitrogen and sometimes sulfur. But these are the main components of proteins: carbon, hydrogen, oxygen, and nitrogen. Sometimes we add to them sulfur. It is again another type of protein. Sometimes we add phosphorus, another type of protein, but these are the main components of protein. Nitrogen must be present. About the nucleic acids, there is hydrogen, nitrogen, and phosphorus. If you want to pronounce them, cho, chon, chomp. What's the difference between these two? Because they are formed of the same elements, then what's the difference? It's the number of the carbon, the number of the hydrogen, and the number of oxygen. In carbohydrates, we have less carbon, so here we have more carbon. In carbohydrates, we have less hydrogen, and here we have more hydrogen. In carbohydrates, we have more oxygen, but in lipid fats, we have less oxygen. That's the difference, only in the number of these elements. And of course, the fat molecules, they are much, much bigger than the carbohydrate molecules. Now, let's go now to the classification, or what are they made of. And by the way, all the organic molecules or the biological molecules, they are large molecules. We call them the macromolecules. Macro means large. So all of them, they are large molecules, and they are formed of basic units, which we call them the monomers. It's not really important to know this word, the monomer, just enough to know that the macromolecules, they are formed of basic units. So let me take at the beginning carbohydrates. These are, of course, macromolecules, and they are formed of basic units. But before I start the basic units, we have to know that carbohydrates, they are either simple sugars or complex sugars. In simple sugars, we have two types. We have monosaccharides, and these are sugars that are formed of one molecule only. Mono means one. Like what? For example, like glucose molecule. This is a monosaccharide. We have also fructose. This is another sugar, monosaccharide. Galactose. These are monosaccharides. If I added glucose to any one of these, they are going to give me the second type of simple sugars, which are the disaccharides. Di, of course, means two. So it means that these sugars, they are formed of two molecules of monosaccharides. It means that monosaccharide is the basic unit for the disaccharide, because if I took any two of them and I added them together, they are given, or I joined them together by a chemical reaction, of course, this will give me a disaccharide. So disaccharide is larger than monosaccharide, and monosaccharide is the basic unit. Like what? For example, like maltose. Maltose is formed of glucose and glucose. It means two molecules of glucose will give me one molecule of maltose. The other example, we have sucrose, and this is the table sugar that we use, and it is formed of glucose together with fructose. The third example is lactose, and this is the sugar of milk. This is formed of glucose and galactose. As you can see, that glucose is common in all the disaccharides because glucose is the simplest molecule of these carbohydrates. And I'm going to give you another hint about glucose now. But let me go at the beginning to the complex sugars. Complex sugars means that they are macromolecules as well, and they are formed of their basic unit. Their basic unit is glucose. So if I have many molecules of glucose, they are going to give me this complex sugars. So examples of complex sugars, we have starch, and it is formed of hundreds or even they may reach thousands of glucose. We have glycogen. It is again formed of thousands of glucose. So what's the difference between both? Starch is always found in plants, never in animals, and glycogen is found in animals. There is a third type, which is cellulose. Cellulose, of course, as we all know, it forms the cell wall of the plant cells. So this is the classification of carbohydrates. Now, why glucose is a main component in everything? And here, here, and here, all these are macromolecules, and glucose is the main component of these macromolecules. Yani, as for the complex sugars, they are formed of glucose only. For the disaccharides, glucose together with another monosaccharide. But why glucose? Now, we know from our previous study that the body needs energy, whether the animal body or a plant body, it needs energy all the time. Yani every now and then, we need an amount of energy. Every part of a second, we need an amount of energy. So energy has to be produced very fast. Now, we have monosaccharides, glucose, we have fructose, and galactose. All of them are monosaccharides. It means that there are sugars that are formed of only one molecule. Why the body takes glucose for its respiration? Because in respiration, we do this. Glucose plus oxygen in the body cells, of course. This will give me carbon dioxide together with water and energy. But why does the body use glucose and not fructose and galactose? Because glucose has the simplest structure. And when the body needs to extract energy, it's going to break down this glucose molecule and extract the energy from it. The body, because it needs every now and then an amount of energy, this means that it has to be fast in extracting this energy. That's why the body refers to the simplest kind of structure of molecule and this is the molecule of glucose, of course. Now, here, why did I write this equation in a word form? Because in the exam, they can ask you to write an equation in the word form, yani either for respiration or for photosynthesis. You must not write any symbols. And sometimes they ask you to write a symbolic equation. So you are going to write it, of course, in another way, like C6H12O6. This is glucose, of course, together with oxygen. This will give me carbon dioxide plus water plus energy. Energy is the only word that is written, but all the rest are in symbolic form. If you wrote in the test, when they ask you about a word form, you wrote glucose plus oxygen, this is wrong. You lose the mark immediately. If you were asked to write a word equation, you have to write this one. If you're asked to write symbolic equation, you have to write this one. And of course, I'm going to balance them, 6 molecules. I just said a word that the body extracts energy from glucose. How does this happen? Now let me take this glucose. As I just said, the main component is carbon. So carbon and hydrogen, of course, because they are organic. This is an organic molecule, but because carbon is more important than hydrogen, because carbon, I have 6 carbon atoms. The carbon atoms are joined together like this, 5 and 6, by bonds. You know the bonds from chemistry, yes?
Yes.
Okay. Which type of bond is this, ya Salman?
Single bond.
Single what?
Yeah.
It has
Single
a name.
bond.
Yes, it has a name.
Covalent.
Covalent, excellent. So here, this single covalent bond is binding the carbon atoms together. Now, where are the hydrogen and oxygen? They are just bound by single covalent bonds again with the carbon. It means that the carbon is the main chain to which are attached the rest of the elements. Now, this bond is a form of energy. Yani it means that the energy is stored in these covalent bonds. So when oxygen comes, these covalent bonds are broken down like this. And because I said that this energy is nothing but stored, these bonds are nothing but stored energy. So if we broke the bonds, so the energy is going to be released. And that's what happens in respiration. There is breaking down of the bonds between the carbon atoms to release the energy that is binding the carbon atoms together. This means that this glucose molecule, although it is a monosaccharide, it is a basic unit, it is a very small molecule compared to the other organic molecules. This glucose molecule is an energy molecule or an energy storing molecule. Why? Because the energy is stored in the bonds that are binding the carbon elements together. That's all that I have to say about L-glucose. What about fructose and galactose? Yes, they are monosaccharides, but their structure is more complex than glucose. If the body needs to use the fructose and galactose, both compounds or both molecules have to enter through some enzymatic reactions to change them to glucose, and at the end, glucose is again used in respiration. Let's go now to the properties of sugars. Before I explain the properties, I'm sorry, I have to mention something about the disaccharides. Now, I said that we have, you have to know, Yani, 3 disaccharides: maltose, sucrose, and galactose. Lactose, lactose. I just said that this maltose is formed of glucose molecule, and another one. طب where is maltose found? It is found in malt. طب what is malt? Malt is the syrup that is extracted from barley. يعني they make from it beer and things like that. There is a kind of sugar called malt, and it has this maltose, or a kind of syrup called malt, which is extracted from barley. About sucrose, it is formed of glucose again, together with fructose. Yes. Now, where is this found? It is found in fruits. Mainly fructose is found in fruits. But what about sucrose? It is found, for example, in sugar cane, from which we extract sugar, and in beetroot. And this sucrose is nothing but our table sugar. About the last one, lactose, it is formed of glucose,
Lactose.
and galactose. Excellent. But where is it found, yes, Salman, the lactose? It is the
Milk.
sugar of milk. Excellent. That's why I wanted to revise this very quickly. But let's go now to the properties of all the simple sugars, which are monosaccharides and disaccharides. All of them, they are, of course, sweet in taste, and all of them, they dissolve in water to form, of course, a solution, a sugary solution. Right. What are, as I just said before, that there are basic units for all the organic molecules. Now, the basic unit of carbohydrates is the glucose. Let's go now to the fats and oils. And as I said before, that their molecules are their atoms, carbon, hydrogen, and oxygen. And these are the large macro molecules. They are organic molecules, yes, because they contain carbon and hydrogen together. But what are the basic units for these fats and oils? We have two kinds. We have fatty acids.
Glycerol.
Yes, very good. And glycerol. These are called the basic units for fats and oils. So if they are added together, they are going to give me these fats and oils. But what about proteins? As I just said, that proteins, they are formed of carbon, hydrogen, oxygen, nitrogen, and
sometimes sulfur.
Sulfur, yes. So what are the basic units بقى هنا يا سلمان? You still remember?
Basic units where?
Excuse me?
What do you mean basic units?
A basic unit means the units that form them.
Carbon, hydrogen, oxygen, nitrogen?
These are the elements.
Oh, okay, then.
The basic unit is the amino acid.
Oh, okay.
Remember here, when I told you that there must be a basic unit that forms the macromolecules. A basic unit means a certain molecule, if I added so many of it, so it gives me a macromolecule. And for every kind of organic molecules or biological molecules, we have their basic units. Yani here in carbohydrates, we have monosaccharides, or mainly glucose. Glucose here in the three of them. So glucose is the basic unit of carbohydrates. Basic units means the units that form the fats and oils, they are fatty acids and glycerol. The units that form proteins, they are amino acids. So what about the nucleic acids, or the DNA and the RNA? They are formed of carbon, hydrogen, oxygen, nitrogen, and phosphorus, and their basic units are called nucleotides. And when I reach explaining or revising a nucleic acid, I'm going to say what are the nucleotides. Now here, because these molecules, carbohydrates, fats, proteins, they are found in our food, that's why they have, in order to investigate them, there are some tests called the food tests. Or how can we test for these biological molecules? Now, the first test is to test for starch, and this is called the iodine test. Iodine test, it is the test for starch. Now, iodine, the color of iodine is yellowish brown. And if I added some drops of iodine on, for example, a piece of potato, because potato contains starch, so this brown color is going to change into blue-black. Never say blue only or black only, blue-black together, because it's very, very...
Acceptable?
Excuse me?
Dark blue.
Dark blue, very dark blue. Never say dark blue as well, because it has a name, blue-black color, okay? Now suppose I added some drops of iodine to a kind of food that does not have starch. Will the color of iodine change?
They remain the same, orange brown.
You never say by it remains the same. It remains unchanged. You have to say it remains yellowish brown. You have to mention what is the color that remains. Yani this is another tip.
The color orange brown.
Yes. Yani this is another hint for the exam. You never say the color does not change or the color stays the same. You have to mention what is this color. It will remain yellowish brown. That's it. Okay? You have to be more accurate in solving or in answering such a question. But what about the food test for sugars, or we call them the reducing sugars? There is a test that is called Benedict's test. This test, it tests for reducing sugars. All the sugars that I just explained, they are reducing except sucrose. It is called non-reducing. But what's the, how can we do this investigation? Here we are going to get a test tube like this, or a boiling tube or whatever. And we take the sugar solution and we add here 1 centimeter cubed or 2 centimeters cubed, it doesn't matter. Then we get another solution which is called Benedict's solution.
Also 2 centimeter cube.
Yes, it must be double.
The same
yes.
amount.
Same volume or double?
Same volume.
Same volume. Okay, some books write this, some books write that. يعني actually طبعا موضوع double ده I say it في الAS because whenever you put more in Benedict, it's going to give you better results.
Yes.
Okay, same amount of Benedict. What's the color of Benedict, ya Salman?
The color is blue.
Blue, very good. So all here, this will be colored in blue. Then this is not enough. We have to put the test tube in.
We need to heat.
To direct heat or in water bath?
In boiling water. In a beaker of boiling water.
Yes, this is called a water bath. And you do not say a beaker of boiling water, you say a water bath because it has a name. And of course, we are going to heat. But so we need to heat this solution to a certain degree. So we have to add a thermometer. And sometimes in paper 6, they come and ask you, why do we need the thermometer? In order to adjust the temperature. So we heat the water in the water bath to about 80 to 85 degrees Celsius. We notice this by the thermometer. We are going to see that this blue color changed into brick red. Excuse me? Excuse me?
I know from my book what I memorized is it goes from clear blue to cloudy green, then yellow orange,
Yes,
and finally to
and
brick red.
finally to brick red. Yes, this is correct. Yani, I was going to tell you that there are intermediate colors here. Okay, from green, of course, or cloudy green, or sometimes we call it earthy green, then yellow, then orange, and finally we reach the brick red, where sometimes they write it rusty red. They are both the same. But this means that there is a reducing sugar in the solution under investigation, and that's why it changed the blue color of Benedict's finally into the brick red. So suppose you made this procedure and the color remained blue. So you say that the sugar is non-reducing sugar. Yes, and the only non-reducing sugar is sucrose. So in positive results, this will be given. In negative results, the color will remain blue. Right. This is the test for sugars. What about the test for proteins? Proteins we do. It is called?
Buret test.
Yes. And in this test, we know the color of this burette. It is blue. And when there is protein, it is going to change into purple or violet, or sometimes we say mauve or lilac or whatever, according to the concentration, if the protein is found. Now, we have another kind of food test, which is the ethanol test, or emulsion test.
For fats and oils.
Yes, this is for fats. So what are we going to do? We are going to get a test tube, and we put in it, for example, some oil, and we put on them ethanol. Ethanol, of course, is an alcohol. So, and we shake vigorously or very hard shaking, of course, after we close the mouth of the tube. Of course, we are going to add this mixture of ethanol and fat, or oil.
We have to add water.
Yes, we are going to add water, and we're going we have to shake it, of course, and we're going to see that water is in the bottom, and above there is a cloudy
emulsion.
Yes, like this. Something looks like a cloud, and this is, of course, the emulsion. But why the emulsion was suspended, was on top of water? Because the oil is less density. Yes, than water. That's why it's hanged above. There is another kind of food test that tests for vitamin C. And this is called the Disaccharides. The tests for vitamin C in, for example, any fruit juice or whatever. Disaccharides is blue in color, dark blue. So we are going to put here in a test tube this Disaccharides solution or reagent. The actual name for Disaccharides, you don't have to know them ولا when you graduate حتى. It's dichlorophenyl indophenol, so it's a very long word just to say the Disaccharides, and that's it. So the idea here is when we add any fruit juice that contains vitamin C on this dark blue Disaccharides, we use the fruit juice and add them drop by drop on Disaccharides, and we shake until finally this blue color will change colorless. If this blue color changed colorless, this means that this fruit juice contains vitamin C. Right. What about the DNA? The structure of the DNA. Now, I told you that DNA, I'm going to take it alone, and I'm going to explain it now. When I said the DNA, again, is an organic molecule that is formed of carbon, hydrogen, oxygen, nitrogen, and phosphorus altogether. Here, this is a macromolecule, so it is formed of basic units, and these basic units are called nucleotides. But before I revise with you the nucleotides and what are they formed of, I have to mention, where is this DNA? DNA is found in the nucleus of the cells. If the cells are eukaryotic or they are true cells having a true nucleus, and if it is a bacterial cell, the DNA is found inside the bacterial cell without a nuclear envelope, of course. So this is the nuclear envelope or the nuclear membrane. We call it an envelope because it's formed of double layer, or 2 layers. This is the nuclear envelope, and we see that there are chromosomes inside, and these chromosomes are formed of DNA. Chromosomes, they contain DNA. The DNA is a basic material in the structure of the chromosome. And each chromosome is a very long molecule of tightly packed DNA. DNA molecule are tightly packed like this. Okay, type. What about the nucleotides? And I just said that the nucleotides, they are the basic elements of, or the basic units of DNA. Now, before I mention anything, I have to say that the DNA molecule is formed of 2 strands, and these strands, they are spiraled around each other like this, forming a shape called the double helix. Yes. The 2 strands, they are spiraled or they are coiled around each other or coiled together to give the double helix form. Each strand of these, it's formed of nucleotides. So I have here nucleotides that form this strand and so on, and nucleotides that form the other strand. Is there a difference? No, all of them are the same. But what is this nucleotide? Now, these nucleotides, they are the basic units. And of course, because the DNA has to be made of a double strand, a double helix, so both strands, they have to be joined together by kinds of bonds like this. Do you know what are the types of bonds here? They are called which bonds? They are not covalent, by the way. You know them? Well, you didn't learn this. These are called...
The bases?
No, not the bases. Both strands, they are joined together or linked together by kinds of bonds that are called hydrogen bonds. The strands are joined together to prevent their separation. طب let us take بقى the nucleotides. What are the nucleotides formed of? The nucleotides, they look like this. I'm going to draw only one of them. Phosphate, because I said carbon, hydrogen, oxygen, and nitrogen and phosphorus. So this is the phosphorus in phosphate. And this is a type of sugar because it is DNA, so it is deoxyribose sugar. Yani DNA is an abbreviation of nucleic acid. So that's why the sugar here is deoxyribose sugar. And here there is a base, nitrogenous base, or a base whose main component is nitrogen. So that's why I said it's formed of carbon, hydrogen, and oxygen of the sugar, nitrogen of the base, phosphorus of the phosphate. Now, this is called, or this structure is called the nucleotide. So what are these bases? The bases, I have 4 of them. I have adenine, and you just write an A, thymine, you just write a T, guanine, and cytosine. Now, if this, because I told you that we have 2 strands of DNA, this is 1 strand, I'm going to separate them بقى. I'm not going to draw them in a double helix, I will separate them in order to show you what do I mean. Now here, I'm going to draw the nucleotides. For example, this is the sugar, this is the phosphate, and this is the base. Suppose it is adenine. Now, this nucleotide is attached to another nucleotide by a certain bond, attached like this. This is the bond like this. And here is another base, let me say guanine. Another nucleotide is attached again to the one above, and this is the base, for example, thymine. Another nucleotide, and so on. Phosphate, for example, here I write adenine. But what about the other strand? Again, it is formed of nucleotides. This is the deoxyribose sugar and phosphate. And this is the base. We always look at one strand. Here, the base was adenine. In front of it on the other strand, it must be thymine. Yes. And this is another nucleotide. So here there is guanine. So on the other strand, there must be cytosine. And here in front of it, there is adenine. Adenine is thymine, and so on. This means that the bases, they are joined together such that, because I told you that both DNA strands, they are joined together. And here the joining is found, or is in between these bases. Adenine is joined to thymine by hydrogen bonds, 2 of them. Guanine is joined always to the cytosine by 3 hydrogen bonds. That's why we have the linkage of the bonding here in between the 2 strands. Okay, but why do we always have the adenine in front of thymine and guanine in front of cytosine? Because these bases, they complete each other. We call them complementary bases. They complete each other. So what does this mean? Suppose adenine looks like this. Suppose. So thymine must have the opposite shape. So if we push them against each other, they will give me a straight line. That's why we say that if this is adenine and this is thymine, thymine is complementary to adenine and adenine is complementary to thymine. The opposite for, and the same, sorry, for the guanine and cytosine. If I have guanine like this, so cytosine must be complementary to it, the opposite shape. Like the puzzle, guanine and cytosine. You have to put the opposite shapes of the complementary shapes together that complete a certain picture. Here we can put them together. If we push them against each other, they will give me a straight line like this. Okay, this is what's meant by complementary bases. And that's all for biological molecules, and let's now go to enzymes. A second. Now, from chemistry, we have known that there is something called a catalyst. Now, what is the catalyst? A catalyst,
molecule that speeds up reactions.
very good, very good. This is a chemical material. And because enzymes, they are catalysts, we that work in a living body, we call them the biological catalysts. Okay, type. I just said that the enzyme, the catalyst is a chemical material. Does this mean that enzymes are chemical materials? Yes, they are. They are proteins or protein in nature. And protein is a chemical material. It is formed of chemicals formed of carbon, hydrogen, oxygen, and nitrogen. This means that enzymes, they are proteins that function as biological catalysts for speeding up the chemical reaction. But how does this happen to speed up the chemical reaction? Now let me give you an example not from biology, from chemistry. Suppose I have a test tube like this, and I put in it some HCl. Dilute, concentrated, whatever, but dilute عادة. And then I add here some iron filings, which are small chips of iron. I'm going to add them. There must be a chemical reaction where the HCl is going to react with the iron to give me ferrous chloride with hydrogen. Type. This is the chemical reaction that will happen. And because hydrogen is going to evolve, we are going to know that the reaction is working when we see the bubbles of hydrogen coming out. Of course, we will never see a ferrous chloride. It's just a liquid. But what we are going to see are the hydrogen bubbles that get out. Sorry. So when I add the iron filings to the HCl, the reaction does not start except after few seconds. Why? The reaction started after few seconds in order to give a chance for the molecules of the iron and the HCl to collide together, to give out, to reach a certain energy that activates the reaction. And we call this energy the activation energy. So there must be a certain amount of energy in any chemical reaction to start the chemical reaction with. And the energy here, because we are not heating, so the energy here is coming from the kinetic energy of collision of the molecules together. Now here, the energy that has to be raised is not very high. And if I needed to raise the temperature to, for example, to heat this test tube, it's okay. I'm going to raise the activation energy a bit in order to start the chemical reaction. But what happens in our cells, animal cells or plant cells? Because there are chemical reactions in our cells and the enzymes are working as catalysts to speed up the chemical reaction. What happens inside our cells? Suppose I have a cell like this, and there must be a chemical reaction here, in any part of the cell. Again, there must be an activation energy in order to start this chemical reaction. Without an enzyme, the cell has to wait until the activation energy is reached. And by the way, in the biological molecules inside the cells, we need to raise the temperature to very high temperature in order to start the chemical reaction. If the cell's temperature was raised very much, the cell will be damaged. So that's why we have to add an enzyme. But what does the enzyme do? Yes, it speeds up the chemical reaction by lowering this activation energy. Let me give you an example. Suppose in order to start a chemical reaction in a cell, suppose that the temperature that must be reached in order to start the chemical reaction is, for example, 80 degrees Celsius in order to start the chemical reaction inside the cell. If the cell reached this very high temperature, it's going to be damaged, destroyed. But if we added an enzyme, so the chemical reaction would start at 37 degrees or 40 degrees. So the chemical reaction will start. So we say that the enzyme lowered the activation energy. That's why the chemical reaction happened fast. Because if the cell waited for the 80 degrees to be reached, it's going to wait for example, an hour in order to raise the temperature to 80 to allow this chemical reaction to start, and the cell is going to be damaged before even it reached the 80. But if we added the enzyme, the chemical reaction will be done immediately, because 37 will be reached very fast. That's why we say that the enzymes, they speed up the chemical, yes, by what? By lowering the activation energy. Right. Because the enzymes, they act as catalysts and they are very important, so they are affected by certain factors. But before I start the certain factors, I have to mention how the enzymes work. Of course, by lowering the activation energy, yes, but the scientists, they try to know how are these proteins responsible for certain chemical reactions. So they introduced a theory which is called the lock and key theory. They said that for every enzyme, there must be a certain substance on which the enzyme will work. It means that if this substance is present, the enzyme is going to work like a lock and a key. Suppose I have a lock like this, or a padlock or whatever, and here is the hole for the key. And I have, for example, a key that looks like this. This key is going to open the lock. But if I have another key that looks like this, it will never open the lock. This is what happens for the enzymes. So if the enzyme is the lock, there is a certain key that's going to allow this lock to work or to open. But any other key, any other shape of the key, never. In the enzymes, we have a certain shape on each enzyme, which is called the active site. Yani suppose this is an enzyme, it has a shape here, and this shape is called the active site. Of course, the shapes are not like that. I'm just giving you this, like a diagram to describe, yes, to describe what I want to say. So for this active site that looks like this, it needs a molecule that has the opposite shape. So if they are fitting together, so the enzyme is going to work. Shaba, yani exactly looking like the bases that I just explained, that for every base, there must be its complementary shape, the same. If I have an enzyme that has an active site like this, this shape must be complementary to the shape of the active site. So whenever there is an active site that has a certain shape, there must be a complementary shape for the molecule that it acts on. If I have another molecule that looks like this, this does not fit here, so the enzyme will not work for this molecule, but it will work for this. Now, the molecules on which the enzyme acts, they are called the substrate. For example, I have different substrates for different enzymes. Let me give you an example from what we have learned now. I just said that sucrose, which is a disaccharide, is formed of, you remember, glucose and what? Can you remember what sucrose is formed of?
Glucose and fructose.
Fructose, very good. Suppose the sucrose molecule looks like this. We say that sucrose is a substrate. Substrate means the molecule on which the enzyme that will fit in a certain enzyme. Suppose the substrate of the sucrose looks like this. Sucrose molecule. So the enzyme has to be of the opposite shape, like this. So only this can fit here. So if this is sucrose, the enzyme is called sucrase. The enzyme always take ASE in the molecule or in the substrate molecule. And if I have maltose, the enzyme is maltase. If I have sucrose, the enzyme is sucrase. If I have protein, the enzyme is protease. Okay, the sucrase enzyme is specific for the sucrose because it has a complementary shape to the substrate. So if I put this enzyme like this, and because all the molecules, they collide together, during collision, this sucrose is going to fit inside the enzyme. In the active site, of course, because this is the active site. It has the shape, okay? If the sucrose fitted in the active site, so we call this enzyme substrate complex, and it's written like this, by the way, complex. Now, because of this fitting or the lock and key fitting, this means that the enzyme is going to work. So working here means to break down the sucrose into its components. So I have here the enzyme after working, and here it divided these into two components where these components are released of the enzyme after they are formed. These are the two components. If they are added together, they are going to return this shape again. Here we say that these are the products. When they are formed, they are released from the enzyme, and the enzyme is kept free, waiting for another substrate molecule to lock in. If you can imagine that the enzymes, they work, for example, the same enzyme, it works for 1 million times every minute. Enzymes are small molecules that we do not need too much of them, because every molecule will work for 1 million times for 1 minute only. It can work the same chemical reaction 1 million times in 1 minute. This is the basic of working of the enzyme. They work by lowering the activation energy. They work by the lock and key method, where the substrate will lock with the active site of the enzyme. So the enzyme starts to work to break down the substrate. Then the products are released, leaving the enzyme free, waiting for another substrate molecule to lock in. Now, because the enzymes, they are chemical in nature, it means that they are affected by certain factors. Now, what are these factors? We have 4 factors in the meantime, طبعا. Factors affecting enzyme activity. The first factor is temperature. The second factor is pH. The third is the substrate concentration. And the fourth factor is the enzyme concentration. Now let's take one by one. The temperature. If I have a graph like this, and they bring all these questions a lot in multiple choice. Now, if I have here the x-axis and here is the y-axis, the x-axis is temperature because I'm checking the effect of temperature of the enzyme. And we will have to write degrees Celsius. Now, we have here the rate of the chemical reaction or the enzyme activity. Let me write it, the enzyme activity. Now here, if I drew this graph and I write here 0, 1, 10, 20, 30, 40, 50, and 60, for example. Then I'm going to see how to draw this curve like this. Like this. Now, this is the highest enzyme activity. At which temperature? I'm going to drop a line with my ruler to see at which temperature. The line reached 40. It means that the highest enzyme activity, it is 40 degrees. I call this 40 the optimum, yes, excellent, optimum temperature. It has to be 40 exactly? No, we have to give a range. Maybe, for example, 37 to, for example, 41, for example. Now, what happens if I lowered the temperature? So the enzyme activity will decrease, but it does not reach the 0 point. If the curve reached the 0 point, this means that the enzyme completely lost its activity. But actually, when we lower the temperature, the activity, yes, it is dropping, but it never reaches the 0. It means that the enzyme activity does not reach the 0, or the enzyme activity will reach its lowest activity. But what if the opposite? If I increased the temperature, as you can see that the decline or the curve is steeper than this one, and it reached the 0 point, it means that the enzyme activity will be lost. طب why بقى? Because
due to high temperature, the enzyme denatures.
Yes, but what's the meaning of to denature? It means that it's going to be damaged.
Yeah.
Okay, can it work again when it is damaged? No. So if I want to return back the temperature to 40, خلاص, the enzyme is damaged, it will not work again. The opposite here, the enzyme reached its least activity. But if we increase the temperature, it's going to work back again. But this is the difference between high temperature and low temperature. So why is it denatured? Because this is a protein, and the protein molecule is going to be damaged by heating. So what is the part of the enzyme that will be damaged? The active site. It will be damaged. Like here, when I said that the active site is the part to which the substrate will join, if I heated this enzyme, the active site will not look like this. Maybe it's going to look like this, like this. It's going to be destroyed. So the substrate will never fit in the active site because the active site is damaged. So this is the first factor. The second factor is the pH. But what is the pH? From chemistry, we say it is the concentration of the hydrogen ions in solution. But here the pH for me, for biology, means that whether the medium is acidic, neutral, or alkaline. And by the way, I want to mention something about the temperature. For every living organism, it has its own optimum temperature for the enzymes. Yani for humans, all our enzymes will work at, yes, 37. This is the mean temperature. For plants, all the enzymes will work at about 27. This means that for every organism, the enzymes for this organism, they have their own temperature to work on. But this is not true in pH. Yani I said here all enzymes, but in pH, no. Some enzymes, they work in acidic media. Some enzymes will work in alkaline, but most of the enzymes, they work in neutral media. Here the neutral is from 7 to 7.2. Alkaline is from 7.2 to 7.8, for example. Acidic is less than 2, pH 2. This is for humans, by the way. Enzyme depends, or its activity depends on its pH as well. Again, there is an optimum pH, but this is somewhat different from temperature. So if I have here the pH, of course, no unit, and I have here the activity of the enzyme, so I'm going to see this curve. So if I wrote here the 0, 1, 2, 3, 4, 5, 6, for example, 7, 8, and so on. If you can see here from this drawing that the optimum pH is هتيجي عند تقريبا 5 point something. For example, now if I lowered, this is of course the optimum pH at which the enzyme works the best. So here, if I lowered the pH, the enzyme will reach 0. And if I increase the pH, the enzyme will reach 0. It means that here this differs from temperature. And whenever we increase or decrease the pH, the enzyme will be denatured. In temperature, no. When I increased only, the enzyme will be denatured. But when I decreased the temperature, the enzyme will reach its least activity. But it's not denatured completely. But what about the substrate concentration and the enzyme concentration? Or the other two factors. Now, if I drew again a curve or a graph for the effect, or the effect of the substrate concentration on the enzyme. And here is the enzyme activity. You're going to see a curve that looks like this. Sorry. Like this. It means that when I am increasing the substrate concentration, yes, the reaction is proceeding until it comes at a certain time where the curve is going to level off. It means that the reaction does not increase anymore. But why does this happen? Suppose I have substrate molecules like this, a lot of them, because I'm explaining the effect of the amount of substrate molecules on the enzyme activity. Suppose I have a lot of substrate molecules and I have only 4 enzyme molecules only. So this molecule is going to be fitted in the enzyme because I have only 4 enzyme molecules, and this is represented by this curve. But what's that? It means that the rest of the molecules, they have to wait until these enzymes work and their products are released, then the others are going to fit in the enzymes. This means that I have a fixed amount of enzymes that are going to work, enzyme molecules that are going to work, and whenever I increase the substrate, no, I have only 4 enzymes. They are going to work until here. But what about if I increased the substrate concentration? No, the activity will not be increased because I do not have enough enzymes for all these molecules. If I have enough enzymes, so the curve is going to go like this. Do you understand this part?
Yes.
Okay. What about the last factor, which is the enzyme concentration? It means that I have, again, a graph. This is the activity, and this is the enzyme concentration. Enzyme concentration, and here is the activity. Now, again, the curve will look like this, the same. And it levels off at a certain time, leveling off. So what does this mean? It means that I have a lot of enzymes. I'm increasing the amount of enzymes, but I do not have enough substrate. For all these molecules, I have, for example, only 3. So these substrates are going to be locked in the enzyme to give me this curve, or this part of the curve. What about the rest of the enzymes? Whenever I increase the enzyme, there is no reaction. So that's why the activity of the enzyme becomes constant, or on the curve it levels off, or we call it that the curve showed a plateau. Plateau is this shape. Either you say leveling off or plateau. They are both correct. These are the factors that affect the enzyme reaction. What are the chemical reactions that the enzyme perform, or the types of chemical reactions? The types of chemical reactions are either catabolic reactions, and I explained this word last time, catabolic reactions, or anabolic reactions. Anabolic reactions. What does catabolic reaction mean? A catabolic reaction means breaking down reaction. Like what I just explained, if you have sucrose, it will be broken into glucose and fructose. What about the anabolic? Anabolic means building up. It means that if I have several amino acids, I'm going to join them together to build up protein. But how does this happen? The same. Suppose I have an enzyme like this, for example, that looks like this. So I have here 2 amino acids that look like this. These are going to join on the active site. So here the enzyme is going to join them. Again, this is an enzyme substrate complex where both molecules will fit here. And then the enzyme will work on them to join them together. So the product will be like this. and here is the enzyme is left free for other amino acids to work on. This is a building up process. I had basic units or small molecules that are joined together to give me a large molecule, which is a building up reaction.